Well, hello students. I hope you all are in good health. You are staying at home and you are taking your studies seriously. Okay, very well. Uh, what we are going to talk about in this video lecture is digestion. We have already covered in the previous sections about the alimentary canal and different digestive glands. In particularly, we covered salivary glands, liver and pancreas. We spoke about their secretion, their composition, and their functions, right? So in this lecture, we are moving to digestion. Now, what is digestion? The first thing is, what is digestion? The process of breaking down complex molecules into simple molecules is called digestion. Okay, so understand, digestion is the process of breaking down a complex molecule into simple molecules, right? Now, to do that, to understand this whole process, you need to understand what is a complex molecule and what are simple molecules, correct? Let's understand. So to do that, what I'm taking here is a starch molecule. Now, as I have said earlier, starch molecule is a polymer of glucose, okay? And when we say polymer, polymer is a chain like of thing, right? So suppose you take glucose and you start joining them end to end, you will end up having a linear chain of glucose molecules, right? Now this complete chain is one molecule. This is one molecule of starch. And now this one molecule is really big, really heavy, and this is an example of complex molecule. Okay, so whenever we talk about polymers, these polymers are complex molecules. In this case, we are talking about starch, which is a polymer of glucose. Correct? Now, what if we break down this starch into glucose? Again, the units which joined combined to form starch, a complex molecule. What if we again break down the starch? If we break down the starch, we will end up having individual small glucose molecule, and these are simpler molecules, right? And this is what digestion is, correct? Breaking down a complex molecule into simpler molecules, correct? Let's see. So this is starch, which is made up of like thousands of glucose molecules. When there is the process of digestion, the process of digestion takes place with the help of enzymes, okay? And here, the enzymes that act on carbohydrates or polysaccharides are known as amylases. Okay, so if you subject starch with amylases, what will happen? It will break the bond joining the two glucose molecules. As a result, the glucose molecules will be freed, and what you will get is simpler glucose molecules. No more large chain or single molecule of starch, right? Now it will be many glucose molecules. So say for example, there is one starch molecule which is made up of 1000 glucose molecules. If it is undergoing complete digestion, what will you get? From one starch molecule, you are going to get 1000 glucose molecules. Okay, let's see. Great. So complex molecule is converted into simpler molecules, and this is what digestion is. And this is happening with the help of enzyme. Okay. Let's take another example. Here I am taking example of protein. Okay. Now, when we are talking about protein, proteins are made up of different amino acids. Okay, these are polymer of amino acids. So again, if you are going to take a protein, say for example, in a protein there are hundreds of amino acids, it's a complex molecule, and you subject them to enzyme. The protein digesting enzymes are known as uh, proteases. Okay, so once there is a protease, it will break down the bond between these two amino acids. As a result, the amino acids will be freed, and a complex molecule will be converted into simpler molecules. Protein will be converted into amino acids. Okay, now what exactly happens during digestion is breaking of bond between the monomers, right? As in case of starch, you can see I'm drawing a red line which depicts the breaking down of this bond between the glucose. Okay, Now here this bond is called glycosidic bond. Okay. Similarly in proteins, the bonds being braked are peptide bonds because these peptide bonds join the two amino acids to form protein or polypeptide. Okay. So this is the process of digestion now. I hope you appreciate it. Okay, digestion, how the digestion works. What do we mean by saying that breaking down of complex molecule into simpler molecules? Okay, similarly, when we talk about breaking down of nucleic acids, what we mean is we are breaking down a polymer of nucleotides into individual nucleotides and its components, and so is the case of the digestion of fat and lipids. Okay, great. Well, when we are talking about digestion, we know that digestion always takes place with the help of enzymes, right? So let's have a look that which enzyme works on which kind of substrate or which kind of food material. Okay. So when we are talking about proteins are broken down or digested by a group of enzymes called proteases. Okay, so proteases work on protein. Proteases enzymes will break down the protein into amino acids. Okay, great. Lipids or fat are acted by lipases. Okay, nucleic acids are acted by nucleases and carbohydrate by amylases. This we have already studied this. Okay, great. Well, now let's see how, where, what is digested, right? Now, for that, I am making here again an elementary canal. You see, great. So this is elementary canal. Now, the process of digestion starts from the buccal cavity. Okay. So do remember the process of digestion starts from buccal cavity, and inside buccal cavity only and only carbohydrates are partially digested by the help of ptyaline enzyme, which is also known as salivary amylase. Okay. Now the food from buccal cavity goes into esophagus, and in esophagus there is no digestion takes place. Okay. Now food comes to stomach. In stomach, only and only proteins are digested with the help of pepsin and renin enzymes, which are secreted by the gastric glands, which are part of gastric juice. Okay. Now, once the food reaches small intestine, now in small intestine, carbohydrate, proteins, fats, nucleic acid, every component of food is digested. Okay. And this digestion takes place by the help of enzymes of pancreatic juice, okay, by the help of bile juice, and by the help of enzymes of intestinal juice. Okay, now the intestinal juice is also known as succus entericus. Now here do remember that I'm not saying enzymes of bile juice because bile juice do not contain any enzyme. Okay, great. So what you see here is the digestion is started into buccal cavity where carbohydrate is partially digested, not completely digested. Okay. In stomach, only proteins are digested, that too partial digestion. In a small intestine, the carbohydrate, protein, fat, and nucleic acid. So remaining everything, whether it was partially digested or undigested, everything is digested in small intestine. Okay, and this happens with the help of enzymes of pancreatic juice and enzymes of intestinal juice, okay, and bile juice. Okay, also in a small intestine the digestion is completed. Okay, so digestion is completed in small intestine. Okay, so if there is a question that where the digestion is completed, the site is small intestine. Now let's see digestion in more detail, right? Let's see how digestion takes place in buccal cavity, then in stomach and small intestine. So one by one, we are going to see in more detail the process of digestion. Very well. So let's start with the digestion in buccal cavity. Okay. Now, as I said, it starts in buccal cavity. In buccal cavity, only carbohydrate is partially digested, not completely. Do remember, only partially. Okay, now this carbohydrate, which is mostly starch, is acted upon by ptyalin or salivary amylase, and it is converted into disaccharides. Okay, now the question arises is, what is disaccharide? Okay, let's see. See, glucose is a sugar, or we also call it as saccharide. Okay, now if there is a polymer of glucose, so there are many glucose in one molecule, as you found in starch, is called polysaccharide. Okay, such molecule is called polysaccharide. Okay, if the polysaccharide is broken down, okay, into small pieces having 3 to 10 units or 3 to 10 sugar molecules, we call it oligosaccharide. If you break into part having only 2 glucose molecules or 2 sugar molecules, we call them disaccharides. Okay, okay, and if you further break down the disaccharide, you will get the individual glucose molecules, and those are called monosaccharides or simple sugars. Getting it? Correct. So what is happening in buccal cavity? With the help of ptyalin or salivary amylase, the polysaccharide is being converted into disaccharides. But not all carbohydrate, not all polysaccharide is converted into disaccharides. Okay. Uh, about only 30% or even less than 30% of carbohydrate is converted into disaccharides in your buccal cavity. Rest remains as it is. Okay, very good. So see, it means what is happening is in buccal cavity, the food is chewed, right, with the help of teeth. Then it is moistened with the help of uh, saliva, and then partially digested with the help of ptyalin enzyme into paste-like structure, and then it is swallowed. Now, this paste-like substance which is swallowed is called bolus. Okay, do remember, it is bolus. So the swallowed food, the swallowed lump of food is called bolus. Great. So this bolus now moves down the esophagus and reaches into stomach. The rest of the digestion takes place into stomach and small intestine. Okay, so remaining part of digestion we are going to see in the coming video lecture. So this is it for this session. Thank you very much and stay safe.